GU ASCO is an educational session. I think over the years it's shifted to start to include more of a more primary results than they used to do. Last year they decided that they would include a lunchtime session, an optional session run by the excellent statistician Steve Goodman, and apparently people queuing out of the door in order to attend. So they decided they were going to put something into the main session this year to provide an educative element about basic clinical trial statistics, and they were kind enough to ask me to do it. So the aim yesterday was really to help people bob back up the surface for their statistical knowledge, not to get into anything new and complicated, but to really give people a sort of refresher of statistical knowledge. So the title they gave me yesterday was Beyond P-Values. It was about clinical and statistical significance, which is a complicated topic to cover in 20 minutes, particularly bearing in mind the basics they're asking me to cover as well. The argument I was trying to make is it's sort of a false dichotomy to pick on those two things. What we're really looking for is something that's clinically important as well as being statistically significant. So by statistically significant, we're trying to get to the idea that this is convincing data that people are looking at, or how convincing is the data in terms of the strength of the evidence that people have collected. But we have to put that in the context of the clinical relevance of the data that people have collected. Are they seeing an effect that would be clinically important? So I drew people this little matrix about clinically important results and statistically convincing results, and suggested that they should be asking themselves a question: how important is the effect that's been determined, and how convinced are they that this is a real effect? And so if the answer is sort of yes and yes, if it's positive to both of those, it should be in sort of this top right-hand corner of this matrix: clinically convincing, statistically convincing, clinically relevant, statistically convincing. And the example I gave was Karim Fizazi's latitude data from 2017, but there's many things that could fit into that box. Sometimes we'll have things that are clinically important. There's a big effect there, or they're seeing a big effect, but it's statistically uncertain. And at the moment, if the p-value is less than 0.05, people have a tendency to think that there's no effect. Well, actually, there might be effect. What we haven't got is convincing evidence that there's an effect. So if they see that, what they should be doing is going away and getting more evidence, or putting it in the context of evidence that we've already got. And then we've also got things that may be statistically convincing, but the effect is too small to be real. And is that good use of evidence that we've gone to, or good use of time and money to target those kind of effects? So I've been involved with Stampede for many years. It's worked out to be a rather remarkable trial. So the concept of the multi-arm, multi-stage approach was really that of our director, Max Palmer, back in the early 2000s. The concept, and it's one I've very much grown up believing in, is if you've got many things that are worthy of looking at, then rather than setting up competing trials or doing things in sequence, you should put them all into one question. And given that we know that most new treatments don't work, we should be using a series of interim analyses to see if we can move away early from things that aren't going to be sufficiently interesting. So that's the basics of the multi-arm, multi-stage approach. And then we've grown that to start to draw in new questions. If you've got something that's important and worth asking, and there's already an ongoing trial, rather than set up the competing trial, you should try and incorporate it. So with the clinical aspects really led originally by Nick James, who's a remarkable chief investigator and a man with boundless energies, and then drawing in other excellent clinical experts, Stampede's gone on to ask a number of questions over the years. So the most recently reported comparison, first result of a comparison from Stampede, was the M1 radiotherapy comparison, where the lead comparison chief investigator was Chris Parker from the Royal Marsden, the Institute of Cancer Research in Sutton in the UK. This was a trial looking at men who had metastatic prostate cancer, who were starting long-term hormone therapy for the first time. Now, for men with localized prostate cancer, we know that generally it's a good idea to give radiotherapy to the prostate. This was now looking at whether for men whose disease has spread elsewhere, should we also be irradiating the prostate? And there was good biological rationale to support that. We recruited just over 2,000 men to the study, so we got lots of power there to assess the question. What we found was that overall, there was no good evidence for an effect to support the use of radiotherapy. There was a smaller Dutch trial called Horadd that reported a few months before Stampede, and they had broken patients down by whether they had high volume metastatic disease or low volume metastatic disease, whether it spread to many places or only spread to a few places. And what they saw was a very substantial effect for men whose prostate cancer had spread a little bit, but no effect for men whose prostate cancer had spread a lot. And so before we looked at the data in Stampede, we pre-specified that we would do that subgroup analysis as well in the patients for whom we could determine metastatic volume, which was the majority of them. So we worked out what power we would have separately for each of those comparisons. And then as well as the overall effect we reported in those patients, really saying that we believe there should be an effect. If there's going to be an effect and it's not in everybody, then it would be in the low volume patients. And that's what we saw, a strong effect on overall survival for the men with low volume metastatic disease. There was no evidence of an effect in men with high volume metastatic disease. And the point estimates we saw in both those groups were almost identical to what Horadd saw. One of the things I went through yesterday was a simple sample size calculation with the audience. I think it's quite helpful for clinicians to remember how these are calculated. So the information that the example I gave was that we imagined a condition where median survival is 4 years. Half the people are going to live longer, half the people are going to live less time than 4 years. And we were going to target a 25% relative improvement, a hazard ratio of 0.75. It's a pretty standard effect that one might look for in cancer trials. Now, to work out how many patients we need, there's a number of characteristics that one's going to put in. So we need to put in our alpha and our beta. Our alpha is our type 1 error. It's effectively a type 1 error is saying that something's going on when nothing is going on. It's kind of a false positive, and our alpha is our sort of acceptable risk of making that type 1 error. And we usually set that as 5% for traditional reasons. A beta is our acceptable risk of making a type 2 error. Type 2 error is the converse of that. That's saying that nothing's going on when actually it is, of missing something, of a false negative. And so we generally take that as slightly more relaxed, 10 or 20%. And the power is just one take away that, so 80 or 90%. These are traditional values from you can pick any values. So if we take the alpha and the beta of say 5% and 20%, and we put them together, I'm sure it's 20% I used, put them together with that hazard ratio we're looking for, what we get is a number of events that we required in order to see an effect. And so the number of events was something like 382. By events we're talking here, that's deaths in this example. So it's miserable. They're recruiting enough patients till you've got 382 deaths. And it's real people, and I hate thinking about it in those terms. But how many patients do you need to recruit in order to do that? Well, that depends on that survival, that effectively the event rate, the rate at which the events are going to come in. The accrual rate, how quickly do you recruit patients and over how long, and how quickly you want an answer. So I gave an example of what if we recruit for 3 years and we want an answer in 5 years. Put that together with 4 years survival to look for those 382 patients, and we needed, I can't remember the number, it's about 900 to 1,000 patients you'd need to recruit at a rate of about 300 patients per year. So that kind of gives us a sense. And then from there, one can play around, one can investigate the impact of, say, looking for a larger effect or for a smaller effect. And if you look for a larger effect, hazard ratio of 0.65, much easier to pick out the signal from the noise, and you'd need far fewer patients. But you've got to think about whether the effect size is plausible. If you look for a smaller effect, it may be more modest. You've got to ask whether it's clinically relevant. But you need many, many more events and therefore many, many more patients. So a hazard ratio of 0.85, a much more modest effect, which translates into improvement in median survival from 4 years to about 4 years and 8 months, would require nearly 3 times the number of events. Was it 4 times the number of events and 3 times the number of patients? So suddenly you go from 900, 1,000 patients to nearly 3,000 patients in 3 years. That's impossible for most people. So thinking about what the implications are, and then being generous when they read people's papers to think about what they've done to try to get to the study that they're then presenting. It's really important in any study that we choose the right outcome measures. Now, we know in many instances that we want to look at overall survival. That's true in cancer trials. It's a very objective outcome measure. and it used to be where prognosis isn't very good, then it's very easy to get to those events miserably, right? Where patients are doing better, then we need to choose different outcome measures. And we need to agree what those outcome measures are. So Chris Sweeney and the ICECAP group have been starting to look at harmonisation of outcome measures. There's also, in prostate cancer and in many, many other disease settings, not just in cancer but beyond, there's a lovely group of work called COMET, which is Core Outcome Measures for Efficacy Trials, led by Paula Williamson. The work was initially funded by the Medical Research Council's network of hubs for trial methodology research, and then got funding from the European Union. Effectively, it's trying to define core outcome sets that should be used to support research, things we should collect for all patients, regardless of what the outcome measures of the trial, to support meta-analysis and to support facilitation of patients. So loads of MD students and PhD students are out doing research, trying to determine core outcome sets that should be collected. And so whenever one is thinking about what the outcome measure is that you should use, you could turn to COMET and look. There's also a really great piece of research from the Delta 2 or the Delta Squared Consortium, first authored with Jonathan Cook from Oxford, and they set out guidance on how to choose a primary outcome measure for a clinical trial. One of the key bits that jumps out to me is choosing an outcome measure that's really important to at least one key stakeholder group, an outcome measure that really makes a meaningful difference with a justifiable improvement for a group. And by a group, I'm thinking that patients are a really important part of that, not just a practitioner's perspective, but the patient perspective as well. What's important? Well, if you think about examples where we may have a potentially clinically important effect, but we're not sure about whether it's statistically convincing overall. I mean, that's really what phase 2 studies are. Phase 2 studies are looking at early outcome measure. We see something that may be clinically important, is statistically convincing within the outcome measures we pick, but we then go off and collect more evidence. So a nice example of something we should potentially explore further is the combination of zoledronic acid and celecoxib that we looked at amongst the original 5 research arms of Stampede. So there, when we looked at our interim outcome measures, it wasn't looking sufficiently interesting, and our data monitoring committee looked at our predefined criteria and said, that's not interesting enough, you should probably stop recruiting. So we stopped recruiting for those patients, and we followed them up. They continued to have their treatment, or most of their treatment. And then we followed up and we reported, the first author with Malcolm Mason, a paper in Journal of Clinical Oncology in 2017. And what we saw for that combination in a subset of metastatic patients was a hazard ratio of 0.78. I think it was 0.78, very close to the hazard ratio of 0.75 we were targeting, which could be a clinically relevant effect. And statistically, there was some evidence that some people might think might be convincing. Personally, we'd stopped early. It's an intriguing result, clinically and statistically. So it's not going to define practice. We wouldn't want to claim a win for it because of the design of the study. But it is the sort of thing people might look at and say, we should go get some more data. Our conclusion of the paper was where people are already using zoledronic acid or other bisphosphonate drugs, then potentially they should go and do studies looking at other COX-2 inhibitors. There are folk within the Stampede trial management group who look and say, actually, we should have another go. Within Stampede, we could look again. We could do another study looking at bisphosphonates and a COX-2 inhibitor and see whether, and collect more information. So whether that would be more believable done by another group or whether it would be done efficiently by putting it back into Stampede is up for discussion. But I think that's a nice example of something that's intriguing and we need to go and get more data.